Hello everyone, welcome to the seventh SOLAS seminar. I'm Yuzo Miyazaki from Institute of Low Temperature Science, Hokkaido University in Japan, who will host today's seminar. So from International Project Office, Lily is absent today. Instead, Yan, our new project officer, will help us. Thanks, Yan, and Chen will also support us. Today, we have almost 130 registrations from 31 countries, regions. At this moment, we have nearly 50 participants. So for all the participants, please be sure that the seminar will be recorded besides some parts of the presentation and available to watch later on the SOLAS YouTube channel. So before we start the seminar, I'll just briefly introduce the framework of the seminar relevant to the SOLAS. I'll skip the detailed introduction of SOLAS, but in the framework of SOLAS, among 5 core themes, this SOLAS seminar is aligned with some of the research questions of core theme 4, interconnections between marine ecosystems, aerosols, and clouds. So actually, the term ice-nucleating particles, INP, is not explicitly shown in this figure, but specifically airborne INPs facilitates the formation of ice in clouds, the amount of which can affect cloud lifetime, precipitation, and radiative properties. Also, as you may know, it has been shown that marine aerosol particles contribute more to ice nucleation at oceanic regions where dust or bioaerosol of terrestrial origin is too sparse to freeze ice efficiently. So among that, sea spray is one of the major global source of atmospheric particles. Although it is unclear to what extent these particles are capable of nucleating ice, particularly in terms of ice nucleation activity of more chemically complex ambient organic matter linked to a wide variety of marine biota. So today's seminar will highlight recent topics on ice nucleating activity of sea spray aerosols, marine microbial community with its possible link to INPs, and ambient INPs in the Arctic Ocean revealed by a wide variety of studies. So all of these advanced topics are featured by keynote presentations given by the 3 excellent speakers. So each has 20 minutes for presentation, and we'll take questions and comments for 10 minutes after each. And we'll have another time for discussion after all the presentations as well. Okay, so let's move on to the first keynote presentation, a holistic and theoretically based sea spray aerosol ice nucleation model, which will be given by Dr. Peter Albert. So Peter is currently working at the Paul Scherrer Institute in Switzerland. He got PhD in marine and atmospheric science from Stony Brook University, right? And
Yep.
His current research includes environmental microreactor for atmospheric chemistry and physics, and chemical mapping of ice-forming particles, and so on. So could you please your presentation if you're ready, Peter?
Sure. I'm really excited to be talking at this SOLAS seminar today. And I'm presenting a little bit of a controversial topic, looking at sea spray aerosol particles and how we model their ice-nucleation. And so one of the largest uncertainties, I would say, for atmospheric science is ice formation in the atmosphere. And really, I have 2 reasons for that. Initially is that there's a lot of different ways that ice can form in the atmosphere, and that a really small number of particles form ice. And so in the top right of this figure, it kind of gives an illustration of homogeneous freezing, where a droplet, like a cloud droplet that's highly dilute, or maybe something that's an aqueous solution, these can freeze at pretty low temperatures. The temperature is here on this y-axis, and a corresponding altitude in the atmosphere. But if you have a foreign particle, this can cause heterogeneous ice nucleation, Immersion freezing can occur in a cloud droplet or in some aerosol particle in concentrated aqueous solution. Condensation freezing, supercooled water drop freezing, and in a way, all of these we have to get some kind of information about and be able to predict these. Yeah? And that makes things a bit complicated. Sea spray aerosol particles in particular, really the uncertainty of how these form ice kind of struck me about 10 years ago from these two papers, and I just show the results of one where they take a kind of medium estimate of ice-nucleating particles from sea spray aerosol, and that's the red colors, and that's the fraction out of the total, which is sea spray and dust at the same time. And so you can kind of see that these red colors here are around the oceans, and maybe a lot of ice-forming particles in the atmosphere are sea spray. But there's also some uncertainties to these estimates, and the low estimate is here, in which sea spray does nothing. And the high estimate is up here, which kind of sea spray is responsible for almost all of the ice-nucleating particles in the atmosphere. And so this was 10 years ago, and those uncertainties are exactly due to what is the aerosol number concentration of these sea spray particles and their efficiency from some parameterizations. And the efficiency of these particles since the last 10 years has really been studied by a lot of groups using a lot of experimental techniques. I just give two examples from Louisa Eich's group and some work by Thornton et al. And Sarah Brooks, which look at various marine phytoplankton species and sea surface microlayer water, quantifying their ice formation normalized to the surface area of particles here. And also some kind of biological insight into how cyanobacterium are living or in their dying phase and the corresponding ability to nucleate ice. And this is normalized to a volume of air that's sampled here. And so there's been a lot of push on this kind of efficiency, trying to see how good sea spray aerosol particles are at forming ice. I think there's two other reasons why ice formation is one of the least understood processes. And that's because you need to be aware that different types of particles can nucleate ice differently. And sea spray has a lot of different salts, and it has a lot of different compounds. It has a lot of biological or biogenic components into it. And that can really make a question, do we need something that's aware of all of these compounds before we start predicting ice? A fourth reason is I think that there's a lack of a universal heterogeneous ice nucleation theory. Basically a prediction from first principles of thermodynamics or statistics in order to describe the rate of freezing or how many can freeze. And so I go through first how I build such a theory that's aerosol aware, such a model that's aerosol aware and theoretically aware. And I do that by taking the first picture that I showed in the talk and flipping it. And so here what I do is I put the temperature on the x-axis, yeah, and I have all of these different ice nucleation mechanisms, and I put the relative humidity or water activity of a aqueous solution or the relative humidity of the air, and I put that on the y-axis shown here. And all the different ice nucleation mechanisms, some are included here, where if you have a particle that is decreasing in temperature because it rises in altitude and expands and cools, you can create cloud drops, and those cloud drops can freeze homogeneously, and that's this blue line right here. And if you have a particle inside, maybe they freeze here at a little bit warmer temperature, a little bit less in relative humidity, and they all freeze here on this red line that's here. And what's real important about these two lines is they're all constructed from an ice melting curve. So this ice melting curve is a thermodynamic relationship, the ice-water equilibrium, and this is an aqueous solution or as a function of pressure. And essentially, one of these shifts in water activity, or this relative humidity, it's a line of constant homogeneous ice nucleation rate, or a line of heterogeneous ice nucleation rate in the case of the foreign particles that are there. And I would assume in this model that I'm presenting here that the rate of freezing is the same if you're anywhere on this line. And there's a little bit of some physical meanings behind this delta AW, and that comes from homogeneous freezing, is that if you have a single value of delta AW here to define a homogeneous freezing line, that really quantifies a temperature and a humidity for any freezing point whatsoever simultaneously. Here's your temperature, and here's your water activity, and this line has the same curvature as the ice melting curve, and any point on this kind of phase diagram can quantify that freezing point. This delta AW is essentially the water activity at freezing when the water compressibility is a maximum. It's also where you have this equilibrium between low and high density amorphous water, in which the low density amorphous water is suspected to be where ice nucleation happens homogeneously in the first place. If you have kind of molecular dynamics simulations, those are predicting this. Those two papers that go into a lot more detail are down here. I don't have much time for this, but really the question is then if you have this homogeneous ice nucleation line, can you make this heterogeneous ice nucleation line? How does it actually look like? What do we do with this? And so I plot essentially that rate of freezing that I just mentioned on the y-axis here. And for homogeneous, that's per volume per time. And for heterogeneous, that's per surface area per second. And we plot that as a function of the delta AW. And the homogeneous is here, very sharp, and the heterogeneous is a little bit more slanted. And this is already the answer. This is for sea spray aerosol particles that I've derived here. And as an example, if we were to try to predict the freezing from just this line, if we take something like 15 micrometers squared per volume of air in terms of the sea spray aerosol surface area concentration, at some water activity, some delta AW, we get, let's say, at 0.17, and we get here the value of 10 per centimeter squared per second. And if we say, hey, we want 10 minutes to nucleate ice, you multiply all these together, and you get that there's one ice nucleating particle per liter of air. Okay, and this delta AW of 0.17, that exactly means if you have something that's like minus 20, then your humidity is 100%, and that's a cloud. So this would be important for maybe mixed phase cloud having one ice nucleating particle per liter. Or if you're more in like a cirrus regime, here you have minus 50 degrees, and the relative humidity here is 80%. And both of these conditions are exactly a delta AW of 0.17. So that's the model. And now I try to tell you that there is some evidence for this model here. So the evidence, really, there's one theoretical approach from kind of basic thermodynamics, which can explain this water activity shift, this delta AW here. And this comes from the series of papers over maybe the last 10 years by Donovan Barahona, which is predicting the ice nucleation rates. Here you have some Gibbs free energy for activation, which is expressed in terms of the heat of fusion, a surface excess of water at an embryo interface that comes from a ice nucleation theory, kind of treating the interface of the embryo as a sort of having physical properties, and there's a geometric lattice. This is also done not only for homogeneous, but heterogeneous ice nucleation. Here, this is exactly the same model where instead of Gibbs free energy, you have chemical potential, constant pressure and volume, but they invoke this kind of water molecular layer on the surface of an ice nucleating particle being more liquid-like or being more ice-like. It's a parameter that's used, and this can be also used to predict the nucleation rate coefficient, which is shown to be in agreement with previous studies of soil particles as well. For a lot more details into this theory, you can go to some of these papers by Donovan. And so really what the next step is, we have some theoretical basis for the nucleation rates and for delta AW. And now how about this aerosol awareness that we have? So this comes from a paper from Christina McCleskey and some Paul DeMott groups, where the ice nucleating particle concentration is a function of temperature measured at a few field sites are not so represented by aerosol unaware parameterization. So here you have some from previous studies here from DeMott and Morrison up on the top, and that maybe you need some kind of specialized sea spray aerosol particle parameterization of this, yeah? But there's a lot of scatter in this data as well. And if you're so you have a specific function as well as an uncertainty that has to be matched and described. This scatter in the data is not only seen for field collected particles as in here, but it's also seen for a single type of particle. So for one single type of particle here, you get a lot of different instruments with a lot of different range of conditions, and you still get about orders of magnitude uncertainty. And the big question is if you have something that is aerosol aware and theoretically aware, can you get back an accurate prediction with an uncertainty that's completely explained? And that's what I tried to do. Essentially, I've looked at sea spray particles in the past with an X-ray microscope. And in order to see if there's really any big chemical differences between particles that do nucleate ice and particles that don't nucleate ice. And what we found using a kind of carbon spectroscopy to get at functional groups that are mapped in individual particles, we found that those that nucleate ice, an example is kind of here in the bottom left, has a lot of organic material surrounding it. That's this green color, and that's basically dominated by oxygenated functional groups, and an example of the green spectra corresponds to these green colors in the maps here. We see some calcium and some inorganic, but the main point is that the particles that don't nucleate ice, which is over here, they look pretty much exactly the same. This technique has also been done for field studies from other places as well. And really, you kind of get this idea that pretty much all particles have this inorganic and organic coatings, and their spectra look pretty similar. I tried to test that using a mesocosm experiment, where I grew phytoplankton in a mesocosm and aerosolized and looked at the same spectra in the water and in the particles. And those that have nucleated ice, they're a lot bigger, but their spectra is almost exactly the same. Here we have COOH, we have peaks here with the same kind of spectral shape. There's some other little differences shown here and here, but the ones that nucleate ice, really those can be found in all of the sea spray particles, during the growth phase, of growing for a day or 12 days. Those are found in the sea surface microlayer, and those are found in the bulk water. And so what we've done is we've taken this model that we have, which is theoretically based. We've taken our aerosol aware kind of mindset and said, hey, maybe every single sea spray particle is an ice-nucleating particle. Doesn't matter. And we've predicted freezing on all of sea spray aerosol particles, and we're able to reproduce previous studies where in the laboratory and in field campaigns. This is taken from Paul DeMott's work back in 2016 in our model applied. This is only for cloud conditions, so 100% relative humidity, but this can also be predicted for relative humidities that are less than 100%. I'm running out of time, so I quickly just go through the last slide here. We've done this in a cloud model where we've cooled down an air mass and modeled ice nucleation. We've done this for immersion freezing, which is pretty strong for a kind of a more warmer temperature clouds. Some vapor ice phase transitions also come in, and which are dominant here at lower temperature clouds that are more cirrus-like. Yeah? So finally, just in my conclusions, here I say that the ice-nucleating particle composition, and this is mapped on single particles, this can be derived from X-ray microscopy and spectroscopy. And this really balances looking at spatial resolution and chemical selectivity in single particles that do and do not nucleate ice. I make this big claim that every sea spray particle is an ice-nucleating particle. And that should make things a little bit easier if we can just get the flux right in terms of how many are out there. The potential for sea spray aerosol ice-nucleation is really for immersion freezing and deposition freezing. I didn't say too much about this, but it's also been published recently. And finally, I would say that that there now exists this aerosol and theoretically aware parameterization. And I think it's really needed to not only explain ice nucleation results, but essentially explain the uncertainties that correspond to it. And ice nucleation theory will do this. That's the end. Thanks very much.
Okay, thank you very much. So we'll take questions and comments for Peter.
Next question. So you say that C-spray aerosol serve as ice nuclei, all of them. Do you know how would that affect if they are covered, for example, with pollution from the ocean or like organics or other stuff? How would that affect the ice-nucleating abilities?
I think that's a great question. In fact, no, I don't know of a really targeted kind of pollution study for sea spray ice nucleation. But of course, this would introduce maybe modifications to the surface of particles that could nucleate ice. It may modify or it may not. Yeah, in my model, basically, I'm assuming nascent sea spray that is from uncontaminated water, let's say. But it's really important to discuss, I would say. Also, what I think is actually really interesting too is not just maybe the water that might be contaminated, but I don't think there's been a real targeted study on maybe atmospherically aged sea spray particles to some degree. So with like heterogeneous chemistry or something like this. This is something I think that would be also very cool to see if there's changes in the ice nucleation ability.
Thank you. Very interesting thought.
That's a great question.
You have a question?
Yes, I have microphone issues apparently. Can you hear me?
Yes.
Yes, okay, good. Thank you so much, Peter. I kind of missed a little bit when you talked about this chamber experiment where you bubbled chlorophyll, or sorry, phytoplankton, is that what you said?
Yeah.
Yeah, did you see the same ice nucleating regardless if you had algae in the water or not? Or you just meant that like when you were bubbling these phytoplankton, all kinds of particles were ice nucleating?
Yeah, so for all of these experiments that I've done, they all had algae growing. So this is different types of algae. Phytoplankton were blue algae, coccolithophore, diatom, and just kind of spiking a natural seawater as well. And yeah, when we would do this, we've never bubbled just an artificial seawater without any kind of organic material in it, because we just thought this is sea spray, this is just sea salt that would dissolve and not nucleate ice. So it's not something that we did not do, but what we did do is just a variety of phytoplankton, and they all grew over a variety of days, let's say. The results were pretty independent of the growth day, since everything was kind of continually growing. We never really had such like a huge death and like a huge spike in ice nucleation or something like this. We used different bubbling techniques as well, and we really saw no difference in the ice nucleation ability.
That's really cool. But I was just thinking, like, does it matter the concentration? Like, does this have to be an area where there's a bloom, or is this like a global average concentration? And I'm just thinking about like global models. Yeah.
Sure. I do think that in terms of like an efficiency, that's normalized to an abundance, like surface area. I would suspect it's the same. That again would need to have more evidence from a very targeted study. But in the surface area ranges that I have seen, I think that I stated that here. So for surface areas between 9 and 1,000 micrometers squared per centimeter cubed of air, particle numbers between 30 and 5,000 with a certain particle area per experiment. Within these range of conditions, this model is valid, I would say. Outside of this range, not so sure.
Yeah. Okay,
But
thank you so much.
I would say abundance is, so if you have a bloom and there's more particles coming out of the ocean or more surface area kind of flux, let's say, then I think, yeah, the abundance can impact the ice formation in such a model.
Yeah, okay,
Do you have
thank you.
evidence for bloom formed and then cloud formed above it or something? You can test that.
Do I have evidence? No, I've never done such a kind of field study where I've looked at kind of blooming things. At least in my small scale mesocosm, if it's the first day of growth or the 10th day of growth or a big spike as the cell concentrations increase or the amount of exudate that's formed increases, I see no sensitivity there in terms of my ice nucleation experiments. But again, that's just the range of my conditions. I think, something that's targeted with blooms would have to look at the ice formation and the water quality and organic content and cell content and things as well. Yeah. The first steps of that is this paper here by Thornton et al. Looked at how biological kind of activity in waters is affecting ice nucleation. And that's something that I could point out to the community, that this recent paper is pretty good to read.
Okay, is there any question? We have questions in the chat. Peter, can you see the chat? How does your theory compare to singular theory of ice nucleation?
How's your theory compare to singular theory of ice nucleation? Sure. So a singular theory is essentially ice forms from a particle at one temperature. Yeah. And there's one active site that's forming ice there. And my theory is completely different. And the only reason is because my theory has time in it, and that is based off of ice nucleation theory. And so if you wait long enough, you get more ice nucleation that occurs. And also it's different because a singular theory, it really is not a theory. There's no theoretical basis for this. You can't derive a kind of approach from thermodynamics or from some kind of quantum mechanics or anything. It's just a kind of normalization. And so the singular theory, I think, is great for intercomparing experiments or intercomparing maybe field studies that have similar instrumentation. But I don't think it's going to be a good predictor when you try to scale experimental results with atmospheric results.
So as a question in the chat, how much does the uncertainty of sea spray production affect the uncertainty in the ice nucleation?
Oh, I would say a lot, because again, the abundance of how many particles are out there is directly proportional to the rate at which freezing occurs. And so if you're wrong by a factor of 5, then your freezing rate will be wrong by a factor of 5. And I think that's a big, huge uncertainty. Absolutely.
Okay, is there any question? Okay, so we'll move on to the next speakers. Thank you, Peter.
You're welcome. Thanks.
So Nama, could you share your slides now? Okay, the next presentation will be given by Nama Langiona. Her title is Marine Bioaerosols, Emissions, Long-Range Transport, Pathogenicity, and Possible Impact on Biogeochemical and IN activity. So Nama is currently an assistant professor at the Environmental Engineering Department at the Technion Institute of Technology, Israel. And she got PhD at Weizmann Institute of Science and stayed at Max Planck Institute and Weizmann Institute of Science as a postdoc. And now she's working at Technion. And her research interest includes transport, survival, and health effects of microorganisms in the outdoor and indoor air. So, Nama, could you start your presentation?
Yes, thank you, Yuzo, for the introduction and for the invitation. And I would like to say it's difficult times here in Israel. Although we have to move on and continue our work, our hearts and prayers are with the victims of this incredible, horrible situation. So I would like to talk today about the marine bioaerosols. As Yuzo said, basically, intuitively, they would be emitted to the atmosphere directly from the marine environment through the bubble bursting mechanism with organics and other mixed phase marine particles. They would then be expected to impact, as we saw in the previous slide, ice nucleation, water, cloud formation. They would transport and might also impact ecology and global biogeochemical cycles. In these two cases I will present today, we aim to track biogeographical patterns and long-range transport of microorganisms over ocean, meaning to see how the local emissions are mixed with long-range transport of microorganisms, and also report on an aerial infection of a bloom-forming phytoplankton through this emission to the atmosphere. What we did in the study, we basically sampled air bioaerosols on a cruise campaign on the Tara Ocean, where we have this apparatus of filters here on the deck, and tubes connected all the way up to the mast, sampling air through vacuum system to those filters. This work was conducted on board by my dear collaborator, Michel Forrest from the Weizmann Institute, basically collecting samples while the boat was moving, like daily sampling and routinely exchanging filters. We had this campaign was 2.5 years, the sampling campaign, but we had in this work explored two main transects, crossing the Atlantic, which considered a more polluted, more anthropogenically affected environment, as well as the West Pacific transect toward Fiji. And you can see here the inlet and the sampling collection. When those filters were collected daily, every 24 hours, we then froze them in liquid nitrogen and shipped them to our lab. There we did a genomic analysis. We extracted DNA, did sequencing, amplicon sequencing of those DNA to know the microbial population composition in the atmosphere, about 30 meters high, compared to the water community composition. What we also did was to quantify specific species to try to identify fluxes of emission. And you can see here in this figure the back trajectory analysis of the air coming to our filter samples in the Atlantic, where you can see more messy routes of the air going from land, also directional change, compared to the Pacific, where mostly air was coming from the main ocean. And these trajectories are about 5 days trajectories backward. And what you can see in this very dense figure is basically the latitudes where we sampled in the Pacific and in the Atlantic. and the composition of the phyla of this bacterial population in the water, in the Pacific water. Each bar represents another sample compared to the Atlantic. And the air samples above it sampled and the community composition of the air. And what you can see just from looking at this, even not knowing which species which, that the cases are different. We don't see the local emission directly represented in the atmosphere. We do see similarity between oceans, between the Pacific and Atlantic transects, with more homogeneous composition of species compared to what we see more varying species community composition in the air samples of both Atlantic and Pacific. Both have some species coming from different sources. And remember, we are talking about transect of sampling air that indicates at least 5 days over the ocean due to these back trajectories, meaning they transport from very long distance over a long time. We think that the reason for this homogeneity in the marine population is due to the higher stability and longer mixing cycles in the ocean compared to the rapid changes of days to weeks in the atmosphere due to winds and instability. And what you can see in this heat map is basically, again, we selected the most 100 most abundant species in those samples in the Pacific air, Atlantic air, Pacific water, and Atlantic water, and basically representing their abundance of the species. And looking in depth into that, we can see marine species are more represented in the atmospheric Pacific compared to the atmospheric Atlantic. And we think that this is due to the dilution effect of pollutant or anthropogenic impact in the Atlantic compared to the more pristine environment of the Pacific. We see also interesting phenomena of selectivity in emission. We think it's selectivity in emission. For example, cyanobacteria like Prochlorococcus, which is a very abundant marine cyanobacteria, is both abundant in the Pacific and Atlantic water, and is more abundant in the Pacific air. While SAR11, which is also a marine microbe, very abundant marine microbe you can see here, is rarely seen in the atmospheric samples, both in the Pacific and the Atlantic. And we think also from previous studies that this relates maybe to the composition, the chemical composition of the outer layer of this bacteria, also maybe to the selectivity impacted by the surface microlayer. But this is something that we have to further investigate. Also, airborne bacteria. You can see here groups of airborne bacteria that are very abundant, that are sourced from terrestrial, dust-related bacteria, but are rarely seen in the marine environment. Specifically, Pseudomonas, which is known to be an ice-nucleating bacteria, is seen a lot in the atmospheric samples, but we can say that it's seen not coming from local marine emission, although from Peter's previous talk, we know also cyanobacteria are known to be ice-nucleating. And this is, I have to emphasize here, this is a genomic study. We don't know anything about viability of this bacteria. So we have to further check if those species are viable, they might be more efficient, as Peter said before, ice-nucleating. When we compare the contribution of differences between environment, we see that the differences between water and air is much more significant compared to the differences in thousands of kilometer difference between the West Pacific and the Atlantic. You can see that species are more endemic to the water compared to the species that are more endemic to the air, while in the comparison between environment, the Pacific and Atlantic, we see much less differences. Although we tend to see more marine-related species more abundant in the Pacific, and I think, again, it's due to the dilution effect in the Atlantic, which is very contaminated environment. Then we did another analysis trying to understand the genomic composition of our samples and how they were found in other databases available, genomic databases found in different environments, for example, terrestrial environment, Pacific water environment, anthropogenic environment. And what you can see here basically is clustering of those environments, our data set based on the associated environment they may come from. So you can see in the Pacific samples that the majority, like about 50%, are associated with marine environment in the air samples, while not a very significant portion also is related to terrestrial environment. While in the Atlantic, the dominant community in the atmosphere is associated with terrestrial and anthropogenic sources. So this is a very significant and interesting phenomenon for us, indicating import of long-range transported bacteria into the marine environment. I would like now to shift to a different study we did. Same boat, the Tara Ocean, but now we did tracking of a bloom, Emiliania Huxley, which is a copper-4 bloom, and we wanted to understand if we can see what is the emission into the atmosphere. Those blooms are known as very beautifully seen from satellite. You can see the chlorophyll signature and also the signature of the organic compound in the shell of those calcified algae. You can see that from satellite. But what is interesting about them, that they are collapse very fast. And they have patches of kilometer-wide patches of these blooms, and they have a simultaneous collapse. Somehow they have a sense of a connection between them that induce collapse of this bloom very fast in scales of hours and very simultaneously. And this led us to the thinking of maybe the collapse, which is induced by pathogen. We have viruses documented to attack this algae and bacteria. This is a vastly investigated area. These figures are from the Valdi lab in the Weizmann Institute, where I did this study. And we thought that those pathogens might be transported through the atmosphere. And that, wanted to check that, and this is why we went to this cruise to the bloom in the territorial water of the French country to try and track pathogen in the atmosphere. So previous studies on pathogen in the atmosphere have been conducted. You can see here that this showing it how they managed to identify genomically the presence of the virus, the pathogenic virus of this algae in the air. They sampled air unfiltered and did the genomic analysis of this virus, but they did not show here a real infection, the viability or the infectivity of this pathogen from the air. In our study, what we did is to track this bloom over the ocean by looking at the signature of the chlorophyll and the satellite images. We then did, we stayed there for 5 days, about 5 days, and tried to stay in the same environment. You can see here the sampling, the dots inside the bloom and outside the bloom. And basically, the setup was built from 3 different sampling instruments. We had an online monitoring instrument that allowed to evaluate the size of the particles and the amount. We had a spot sampler, which is very good for airborne microbial sampling. It samples into a liquid media, and then we store those samples in 4 degrees and ship them back to the lab to do the analysis. And also filter analysis for the genomic and chemical composition study. In the lab, in a nutshell, what we did was to infect basically healthy culture of this algae and to try and see if there is any infection. We identified, I will let you know that we identified pathogen in this method coming from the air. They are marine originated pathogen. We then did quantification of the genes of those species to try to identify fluxes of emission to the atmosphere. So let me show you in details. You can see here the composition through SEM EDS analysis of these filters that's showing mostly marine contribution of these samples to the air. From the infected culture that we had 2 samples identified to be infected. We isolated the bacteria of these infective cultures and identified 5 different bacteria species. We then reinfected healthy culture with each of those pathogens. And one of those was the one that was infective. We identified Rosovaris nobilhybins as the pathogenic bacteria infected this algae. And you can see here cultures of this algae with controls and antibiotics. And without antibiotics, this culture just died. We then wanted to make sure this bacteria did not originate from the culture itself, from the algae culture microbiome. So what we did was to extract DNA, and we did the 16S amplicon sequencing, similarly as we did in the previous study, of those air filters, but also of the different cultures we infected. And what you can see is that the infected cultures has a very different microbial composition compared to what we see in the air samples. And specifically, when we looked for the isolated bacteria, we could not identify them in these cultures. We did find them in the air samples. Next, what we did, we quantified the quantity of genomic signature of the pathogenic bacteria compared to the algae, this Emiliania Huxley. And what we see is that there are about 4 orders of magnitude difference in the concentration of those bacteria, in much higher concentration compared to what we see in the algae, which makes sense. And it is a very important thing to note also for IN studies. Algae are much bigger than bacteria. If they will be emitted to the atmosphere, they will be emitted as fragments and very rarely as units of cells. This is a very big 10 microns size algae that is rarely emitted to the atmosphere. We think that what we saw in our samples was debris of this algae, but we couldn't find them in the SEM, for example, as a whole intact algae. So basically, that also means that this bacteria will be emitted in much higher concentration to the atmosphere, most likely single cell, not attached to the algae. And we saw higher concentration of those. We still have to figure out how long can they survive in the air after they are emitted. We had luck. Basically, in our sampling, we managed to sample viable bacteria, but we don't know how long and when they were emitted to the air before we sampled them. We also don't know yet what is their infection range, how far they will get in order to get to infect another patch of bloom. So this is something we currently try to understand. And let me wrap this up as a take-home message. We in these 2 studies we saw the bioaerosol population over the ocean is very diverse and very influenced by terrestrial and long-range transport bacteria, but also from local marine population. We still don't know what is the role of the sea surface microlayer in the different emission efficiencies of the different microbes and how that differ between the Atlantic and Pacific Ocean. We ask also what is the microbial viability. We don't know that from this study. And what is the contribution of the atmospheric marine boundary layer bacteria to the global cycle? That would be inferred from understanding viability and bioactivity of those microbes. In the second study, we saw living airborne bacteria that were sampled above algal bloom in the Atlantic Ocean, and they exhibited pathogenic activity and was able to infect healthy culture in the lab. And here we want to know how long can those microbes survive and proliferate in the atmosphere, and how efficient and how far they can reach to infect other patches of cells. We also ask, what is the other bioactivities exhibited under such conditions? If they are emitted, as Peter explained, above a bloom of Amelia Huxley, those bacteria might be also actively involved in water cycle and biogeochemical cycles that we see. Thank you for your attention. I would like to also acknowledge the Tara Ocean Consortium and Michel Flores, Inbal Nussbaum, Ilan Koren, and Asaf Vardi that were involved in these studies.
Thank you very much, Nama. So we'll take questions and comments.
Hello, can you hear me?
Yes, please.
Oh, hi. So, okay, good. So this is Ben Murray from the University of Leeds. So thank you, Nama. I like your ship. That's really nice. One question I had was and sorry if I missed this, was what concentration are those bacteria in the atmosphere? So bacterial cells per unit volume of the atmosphere.
Can you hear me? I feel maybe I'll stop sharing. Can you hear me?
Yes.
So what we identify from the qPCR studies, we find them in microgram per milliliter per liter. Sorry, I have to check the values. But for example, in the air sample that we took from above the bloom, we found them in a concentration of 10 to the -6 microgram per milliliter. Not microgram per milliliter. Sorry, cells per milliliter, which is not a lot. It's much less than we see in the water. It's very low.
Yeah. So that's cells per milliliter of air. 10 to the minus.
Let me check first. I think we did the conversion because in the water is per milliliter, but in the air is per liter of air. But I have to check values. So it's about 6 orders of magnitudes different between the water and the air, what we find in general. I don't want to say something about the units because we did the conversion between liters of air to centimeter, I think, but don't want to say. I think it's a 6 orders of magnitudes difference between them.
Okay. How large are these bacterial cells? What size are they? Micron
or 2.
1 or 2 micron. Okay.
Yeah.
Okay, so they're the top end of the accumulation range. So they'll have a fairly long lifetime in the atmosphere just with respect to sedimentation. What is that about?
We don't know about if they survive there, if they are viable, but you say about ice-nucleating activity, I guess.
In terms of ice nucleation, I don't care if they're alive or dead.
Yeah.
All right, thank you very much.
Sure. Yeah.
So Peter?
Yeah, I'm wondering about the DNA analysis. I guess if a cell is lysed, that DNA is released in water, but is there DNA also in the exudate from a living cell that hasn't lysed yet?
So DNA is considered a very stable molecule. If you check RNA, for example, it will be degraded in very fast. It depends if it's in the water or in the air, but DNA, we find DNA survive, even if the cell degrades or dead, the DNA survives quite good in dry conditions. So in the air, for example, or on our filters, they will be preserved quite good. If they even sampled for 24 hours, they will be preserved there. We did some studies on that, which allows us to really understand the genomic community composition. Yeah.
Yeah, I guess my question is more is DNA kind of released into the water, even so a cell is not lysed or fragmented,
For
just
sure.
in the regular old lifestyle of
Yeah, in the
this.
water, I expect it will be degraded faster than in the air, as we have there a lot of bioactivity, but still it releases also to the water.
Okay, there is a question in the chat. Are virus and bacteria in separate aerosols or virus attached to bacteria in aerosol? From Son Meo Fan.
It's really hard to say. For that, we need to, so they expect to be different entities, but when they emit to the atmosphere, they might be mixed and attached to each other. Depends on their density, basically, in the water. If they are very dense and in an infected bloom, they will be very dense. They might be linked to each other. We saw both viruses and bacteria in our samples. We couldn't identify infective virus in this case, but it doesn't mean they aren't.
Is there any other question? So in terms of community composition in Pacific water you have shown, have you got any data of community composition in the eastern or northern Pacific, especially equatorial or subarctic regions, both of which are, how to say, high nutrient, low chlorophyll regions where the surface ocean dynamics is largely different from that in the western Pacific? So do you have any data with some idea?
We didn't analyze. It's a very vast, major pool of samples that we have. We had to select. So in this case, we selected those transects that we didn't have stops, because this boat, the main idea of the boat was to investigate islands. So when we had transect with islands, we sampled, but this is more influenced by the stopping in the island. So we selected specifically those specific transects that were continuously traveling. I know that there are some crews now in the Arctic from this ship from the Tara that is now investigating also the atmospheric microbiome there. I know there are some studies from the southern part from Australia and the Arctic doing some studies there, but there it was, the sampling was, the apparatus sampling was located inland and sampling the air coming from the ocean. So it's a bit different. Yeah, it's definitely an interesting phenomenon to be investigated. Thank you.
Okay, thank you. So there's also another questions. Now, can you see the chat?
You mentioned that it is
the one
not
from Irina mentioned that it is an open question. How long can microbes survive in the air? Do you think that transport with moist air masses, including liquid clouds, helps microbes survival or over longer distances?
I think yes. I think yeah, and this is why we try to track them and sample them and try to understand distribution. Certainly, clouds would be a nice vector for them to travel in, unless it's you know, temperature-wise, I'm not sure. In the sense of their bioactivity, I'm not sure, but I know there are some studies from Pierre Amato. They do these studies of ice-nucleating bacteria in clouds, and they managed to see some very interesting phenomena through looking at the RNA level. So definitely very interesting topic.
Okay, is there any other questions? It's okay. Okay, so we'll move on. Thank you, Nama. Thank you very much. Okay, so in the remaining time of this seminar, we'll take additional questions, comments for each of speakers or for all. So is there any overall questions or specific to someone? Or do three speakers have questions, comments for each other?
From my side, it was very interesting the finding of these bioaerosols from Tobo's talk. I was thinking, and very initially, from the perspective of the microbiology, he said it might be fungal spores, but it also could be ciliates and single cells. It looks like that in one of the slides. So that if there is any contribution I could give on that, I'll be happy to.
Peter, do you have any comments?
As of now, no.
So Itaka, Tobo-san, do you have any comments or questions?
I have a question to Nama. So do you analyze DNA only for bacteria, or do you also use other approach like 18S or ITS?
Yeah, we have done some analysis on the 18S, but yeah, this data is still raw and not analyzed yet. We do find much more diversity in the bacterial domain, but certainly would be very interesting to also get into that. We plan to do that in the future for sure.
Okay, that's interesting.
So according to the NAMA's presentation, NAMA showed that aerosolization fraction of bacteria is substantially higher than that of algae, right? So in terms of their IN efficiency, Peter, do you have any comments on that, how to say, difference in the production of sea spray?
Yeah, I guess my only comment would be that for the fragments, phytoplankton and whole bacteria cells, for ice nucleation purposes, I think there was a paper from out of the Dan Sitzo group, which talked about some prochlorococcus bacteria, I believe, saying that these were also highly efficient ice nucleating particles. And so, yeah, I could imagine that that if there's a larger bacterial abundance, then yeah, these would have a more impact on the concentrations of ice nucleating particles. I would agree with this.
Again, we do find some debris, but intact cells are much less abundant due to the size, I presume, and the density. So they are much less suspended in the air for longer times. The algae. So cyanobacteria would be a good candidate due to our findings, as well as the other bacteria. Yeah.
Okay, for the participants, is there any other questions, comments? Okay, so we will close this seminar, and I would like to thank all the speakers again and participants, as well as Yan and Shen from IPO. And thank you all again to attend this seminar, and see you next time.